If you are confused about this new technology that is called CRISPR and have questions like where it comes from, how it works in humans, who discovered it, and what are its applications, then this video will help clear your concept about it. So let's begin. What if you can make specific changes in the DNA of human, animal, and plants? Yes, this is possible by a breakthrough technology called CRISPR. CRISPR, it is a powerful genome editing tool which is much faster, cheaper, and efficient. It helps scientists to add, delete, or alter the DNA sequences at particular locations in the genome. CRISPR stands for clustered, regularly interspaced short palindromic repeats. Now, what are the applications of CRISPR? Scientists can edit the genes of crop to make them tastier, healthier, pathogen resistant, and more robust to environmental conditions. They could also remove HIV genes from the infected human cells. Malaria blocking genes can be added into the mosquito. But it is important to note that currently there is no CRISPR edited product yet approved by FDA. Let me tell you a surprising fact that we actually borrowed CRISPR from bacteria and archea, which uses CRISPR as an immune system to defend themselves against the bacteriophage. We know that when bacteriophage attacks bacteria, it injects its DNA in it, then replicates, causing the cell to rupture, resulting in the bursting of cell. Scientists find a series of short repeating DNA with spacers sitting in between them. In 2000s, scientists found that the spacer DNA matches perfectly with the viral DNA. They also identified a number of genes associated with CRISPR. So these are the CRISPR associated or the Cas genes. These Cas genes will make Cas proteins. That's going to be helicases that unwind the DNA and nucleases which cuts the DNA. Let's say this is a bacteria. When virus invades it, the CRISPR DNA produces two types of small RNA called the CRISPR RNA written as cRNA and a tracer RNA. The Cas gene produces an enzyme called Cas9 which is a nuclease. Now the Cas9 enzyme plus the RNAs form a complex that cuts the DNA of the invading virus, thus protecting the bacteria from infection. Scientists then thought if they could create CRISPR in lab in order to alter the DNA sequences in other species. So for that they create a piece of RNA with a guide sequence which is complementary to a target pair of DNA in the host genome. In other words, if the DNA has a sequence which reads 5' GG CT AT 3', then RNA guide sequence is exactly the opposite. That would be 3' CC GA UT 5'. And one thing is important to notice here is that there the U is for uracil because the guide sequence is made of RNA, not of DNA. Then the Cas9 protein attaches to the RNA. Cas9 and guide RNA then binds to the target DNA sequence in the host genome. The Cas9 RNA complex then make a double strand cut in the genomic DNA and an alternative piece of DNA is inserted in that spot. Let me explain you with an example. Let's say there is a girl named Susan. She has sickle cell disease caused by a single DNA letter mutation. In the lab, scientists create a strand of guide RNA that matches the mutated DNA sequence. Now, then, Cas9 enzyme molecular Caesar is added to the RNA mix. Then, Susan receives an injection of RNA plus Cas9 mix. Then, guide RNA identifies the mutated DNA that causes the sickle cell anemia. Then, Cas9 cut out the affected sequence. Scientists insert a healthy DNA sequence to replace the clipped sequence. Lastly, if all goes smooth, Susan no longer suffer from sickle cell anemia and result in a new healthy DNA strand. Feng Chang, who redirected the CRISPR focus from bacteria to eukaryotic cell, described CRISPR using a helpful nursery rhyme. We can imagine a certain DNA sequence that is found in this way. Twinkle, twinkle, big star. That should actually be twinkle, twinkle, little star. So, in this process, first step, the guide RNA locates the error or the mutation, that is, in this case, the word big. Then, in the second step, the Cas9 enzyme makes the break before and after the word big. In the last step, a bioengineered vehicle inserts a correct piece of DNA, in this case, the word little. This timeline presents the concise history and the current contributions of the scientists who pushed this field forward. It all started in 1987 when a scientist named Yoshizio Ishino discovered the repeated sequences of DNA. Then, Francisco Mojico was the first researcher to characterize what is now called a CRISPR locus. In 2000, he named these sequences as short, regularly spaced repeats. Then, in 2002, he renamed it as CRISPR. Cas genes were also discovered. In 2005, spacer sequences in the bacteria and archea were found highly homologous to the viral DNA. Then, in 2012, two scientists named Jennifer Doudna and Emmanuel Charpentier showed CRISPR can be used to edit the human cells outside the body. This finding is now considered as one of the most significant discoveries in the history of biology. Then, in 2014, a scientist named Feng Zhang successfully adapted CRISPR-Cas9 for genome editing in the eukaryotic cells, including the human cells. Then, in 2015, a scientist named Zhenjue Heng and his colleague used CRISPR to modify the non-viable human embryo. Although his paper was rejected by a top Western journal on the grounds that it did not follow the ethics. Then, in 2018, He Jiankui claims to have made the world's first gene-edited babies. His research was also condemned by the scientific community on the ethical grounds. Hope you find the video helpful.